Live by Dr. Pat, leading innovation in fertility excellence.
Imagine you are sitting in this sterile, perfectly temperature-controlled laboratory, right? And you are staring down the lens of a high-powered microscope. On the glass slide in front of you is a microscopic cluster of, well, just 8 cells.
Yeah, just 8 tiny cells.
Exactly. And you have to decide, based almost entirely on what you are seeing, if this tiny cluster has the potential to become a living, breathing human being, or, if it goes into the medical waste bin.
It really is an immense responsibility. I mean, for the patients who are just waiting desperately for a phone call from that clinic, the grade assigned to those cells basically dictates their entire emotional state, not to mention their future.
Yeah, that is the daily high-stakes reality of an IVF embryologist. And, well, welcome to the deep dive, because today we are taking you straight into that laboratory.
We really are.
We've gathered some fascinating sources for you, specifically this comprehensive chronological review of how we classify human embryos, alongside a major new clinical update. It's built around the ESHRE and Alpha 2025 Istanbul Consensus.
Which is a huge deal in the field right now.
Right. So our mission today is to take you on the exact 7-day chronological journey of a human embryo in vitro. We are going to demystify the grading systems at each stage, explain the wild biology behind them, and reveal a massive recent plot twist in what actually makes an embryo successful.
Because, for decades, picking the best embryo was essentially an art form. It was this subjective visual guess.
Mm-hmm.
But today, it is a ruthless, data-driven science.
Okay, let's unpack this. I mean, how do you even begin to grade a potential human life just 16 hours after fertilization?
Right. Well, to understand that, we have to start at the literal beginning, which is day 1. The window is roughly 16 to 18 hours post-insemination. The embryo at this stage is called a zygote, or sometimes a pronuclear embryo.
Okay.
And at this very first stage, the primary goal is simply to confirm that normal fertilization actually happened. So the embryologist is looking for 2 distinct circles inside the cell, which are the pronuclei.
And one of those is the package of DNA from the maternal side.
Right. Exactly. 1 maternal, 1 paternal. And they also look for something called the extrusion of a second polar body, which is basically the egg throwing out its leftover unnecessary genetic material so it has exactly the right amount of DNA to merge with the sperm.
The sources mentioned that historically this was the moment where embryologists became completely obsessed with these tiny microscopic details. There's a lot of focus on these things called nucleolar precursor bodies, or NPBs.
Oh, yes. The NPB obsession.
From what I gather, these are like tiny structures inside those pronuclei. Can we break that down? Are we basically talking about the cellular factories that build proteins?
That is actually a great way to look at it.
Yeah.
NPBs are essential for ribosome biogenesis, so building those protein factories and organizing the chromatin, which is the raw, unspooled DNA.
Got it.
And back in the late 90s, researchers thought these little factories held the absolute secret to embryo success. There was a very famous system introduced in 1999 by Tesarik and Greco.
Right, the 0 to 5 patterns.
Yeah, they categorize zygotes into these rigid patterns from 0 to 5 based entirely on the size, number, and distribution of those NPBs. If the factories were perfectly equal in size and lined up right at the junction where the maternal and paternal pronuclei met.
They called a pattern 0, right?
Exactly, pattern 0. That was considered a golden ticket. But if you had fewer than 3 of them, a pattern 4, it was labeled an absolute failure.
Wow, and researchers just kept drilling down into this, didn't they? You had Scott's Z-score, which graded from perfect harmony down to severe dysfunction if the pronuclei were physically separated.
They really did.
And then you had Gorello's system, which got so incredibly granular it measured the exact angles of the polar bodies, and it looked at a cytoplasmic halo, which is apparently the mitochondria rushing to the center of the cell to power the upcoming division.
Yeah, a massive influx of energy.
It all sounds mathematically pristine, but then the sources introduced this massive plot twist from the new 2025 Istanbul Consensus. They're officially telling clinics around the world to completely stop grading embryos this way. It's just been deprecated.
Completely thrown out. What's fascinating here is that we realized we were grading dynamic processes as if they were static.
Wait, so they were grading all these moving, dynamic cellular parts based on a single look? That feels like trying to review a chaotic ballet by looking at one still photograph. I mean, you might catch a principal dancer mid-fall and assume they are terrible.
That is a perfect analogy. And it might just be a blurry photograph, too, because pre-implantation development is so kinetic. The formation of that mitochondrial halo, the polarization of those protein factories, the fading of the pronuclei as the DNA finally merges.
They happen so fast.
Right. They're fleeting, transient events. If an embryologist looks through the microscope an hour too early or an hour too late, they catch the dancer mid-leap. The embryo might look like a catastrophic pattern 4, but in reality, it is just transitioning normally.
Which means, for patients, a clinic might have historically discarded a perfectly viable embryo just because they looked at it at, like, 2 p.m. instead of 3 p.m.?
Yeah, that is the tragic reality the 2025 Istanbul Consensus recognized. By over-scrutinizing these day one snapshots, we were throwing away potential life.
That is wild.
So the new global mandate is incredibly simple. You just confirm that there are 2 pronuclei and 2 polar bodies, proving fertilization worked, and then move on. Don't grade it. Let it grow.
Okay, so the biology moves forward to days 2 and 3. This is the cleavage stage. And this phase is mechanically really strange. The embryo is dividing, so 1 cell becomes 2, 2 becomes 4, 4 becomes 8.
But it's entirely encased.
Right. It's trapped in this rigid microscopic shell called the zona pellucida. So the total volume of the embryo doesn't actually grow at all. The cells just have to keep splitting into smaller and smaller pieces.
They're called reductive divisions, yes.
Yeah.
You start with one massive cell and you carve it up. At 44 hours, which is day 2, you want to see exactly 4 cells. At 68 hours, day 3, you want exactly 8 cells.
And because day 1 proved too fleeting to grade, I assume this cleavage stage became the new primary battleground for selecting the best embryo.
Exactly. The foundational grading system here was built by Lucinda Wieck, and it relied heavily on 2 things. First, the symmetry of the individual cells, which we call blastomeres, and second, a visual estimation of something called fragmentation.
Let's talk about fragmentation because the sources bring it up constantly. Biologically, this is essentially cellular crumbs, right? Little bits of cytoplasm that break off during cell division but don't contain any DNA.
That's a good way to describe it, yeah.
Yeah.
Under the old Wieck criteria, a perfect grade 1 embryo had 0 crumbs.
But the sources highlight a massive pitfall here. Humans are terrible at eyeballing volume. If 2 highly trained embryologists look at the exact same embryo, their agreement on how much fragmentation exists is just notoriously poor.
It is a profound vulnerability in the lab. Imagine looking at a 3-dimensional sphere under a microscope and trying to guess if those scattered crumbs take up 15% of the total volume or 25%.
Yeah.
I would definitely get that wrong.
It is highly subjective. So different global organizations desperately tried to fix this. In the U.S., the Society for Assisted Reproductive Technology, SART, decided to decouple cell number from fragmentation entirely.
Oh, interesting.
Yeah, they recognized a fundamental biological truth. An embryo that has reached the proper 8-cell milestone on day 3, even if it has 15% fragmentation, is inherently more viable than a pristine, crumb-free embryo that has only managed to reach 4 cells by day 3.
Because that pristine 4-cell embryo is just developing way too slowly to be healthy, right?
Exactly the issue. Meanwhile, a Spanish group known as ASABIR took a totally different angle. They instituted a massive penalty for something called multinucleation.
What does that mean?
So normally when a cell divides, it copies its DNA into one central library, the nucleus. But if you see 2 or 3 nuclei in a single cell, it means that copying process was completely chaotic. The chromosomes scattered.
Yikes. So Essebjorn just drops those embryos down.
Immediately to a category D, because the clinical data shows pregnancy odds plummeting from around 44% to 23% if multinucleation is present.
Wow. And over in the UK, the NUA system tried to solve the eyeballing problem by introducing stage-specific volumetric grading. Basically, they forced embryologists to measure the cells against strict mathematical volume expectations.
Right.
So instead of, cooking by feel and guessing how many crumbs there are, they are essentially forcing the lab to finally start using precise measuring cups.
That's it. It anchors the visual grade to physical reality. And this connects to a broader concept called the embryonic development rate. Timing is just as critical as structure here.
Because there's a strict bell curve, right?
Exactly. Dividing too slowly is obviously bad, but dividing too rapidly, say an embryo racing to 12 cells by day 3, is just as detrimental. Both extremes correlate heavily with metabolic distress and genetic abnormalities. The embryo has to hit the exact right cell count, at the right physical volume, at the right time.
Which brings us to a really tense moment in the journey, which is day 4. The embryo is about 92 hours old, and it hits the morula stage. The sources call this the neglected transfer day. It's essentially a diagnostic black box that used to terrify embryologists.
It really did. It's a very difficult stage to assess visually.
Right, because biologically what is happening is that the mother's initial genetic instructions finally run out. Panic sets in, and the cells have to turn on their own DNA to survive. It's called embryonic genome activation.
Yes, and structurally, they start producing a sticky protein called E-cadherin. They flatten against each other, and they merge into a single, cohesive mass.
You literally can't count the individual cells anymore.
Actually not. The visual markers embryologists relied on for days 2 and 3 completely vanish. But this genome activation is arguably the most critical hurdle in pre-implantation development. The embryo is finally taking the wheel.
That makes sense.
Because it's so important, specialized systems eventually emerged, like the Tau system, that graded embryos based on the percentage of cells that successfully compacted into that mass. And later research proved that if you assess this compaction properly, transferring an embryo on day 4 can be just as successful as waiting for day 5.
But the new 2025 Istanbul Consensus issued some pretty severe warnings for day 4. They flagged 2 major red flags. One is massive vacuolization, where the embryo fills with these toxic lipid droplets, which basically signals the cells are digesting themselves from the inside out.
Yes, a clear sign of impending cell death.
And the other red flag is a failure to fully compact, which honestly makes sense. If compaction requires the embryo to turn on its own DNA, it's basically a cellular trust fall. Every single cell has to participate.
I like that, a cellular trust fall.
Taking that further, if some cells fail to turn on their DNA, the healthy cells just exclude them from the mass. This is kind of like a ruthless corporate merger where the underperforming departments just get walled off and left behind to die.
It is a ruthless but completely necessary mechanism. When we see a partially compacted morula with excluded cells, we are watching biology perform its own quality control.
Oh, wow.
The cells that failed genome activation couldn't produce that sticky E-cadherin, so the healthy part of the embryo recognizes the failure, marginalizes them, and leaves them to undergo apoptosis, which is programmed cell death.
So the embryo is essentially doing the heavy lifting for the embryologist.
Exactly. For a long time, the field avoided day 4 because it was hard to look at. But assessing that compaction process is actually a brilliant, non-invasive way to visually check the genetic integrity of the embryo.
Okay, so if the embryo survives that day 4 trust fall, it arrives at the final destination of the IVF lab, which is days 5 to 7, the blastocyst stage. And this is where the absolute biggest paradigm shift in modern embryology takes place.
It really is a massive shift.
Right. So a healthy blastocyst has 2 distinct parts. You have the inner cell mass, or ICM, which is a clump of cells that will eventually become the fetus. And you have the trophectoderm, or TE, which is a sphere of outer cells that pumps fluid into the embryo and will eventually become the placenta.
Yes, those 2 distinct lineages are fully formed now.
The global standard for grading this is the Gardner scale. It scores expansion from 1 to 6 and then gives a letter grade for the ICM and a letter grade for the TE. So like a 4AA is a beautifully expanded embryo with a tightly packed fetal mass and a strong placental layer.
And for decades, the entire field operated on a very logical assumption. The inner cell mass is the most important part because, well, it becomes the baby.
Right, obviously.
Old grading matrices would automatically label an embryo as poor if the ICM looked weak, regardless of how robust that outer trophectoderm was. The fetal precursor was everything.
Which makes complete intuitive sense. But then the 2023 ACBR decision tree completely upends this. They analyzed the data and found that the trophectoderm, that outer layer, is actually the single strongest independent predictor of a live birth.
Yeah.
Meaning a blastocyst with a mediocre fetal mass but a stellar outer layer frequently outperforms an embryo with a great fetal mass but a weak outer layer. This is wild to me. We always assumed the VIP, the fetus, was the most important thing. But the trophectoderm is essentially the bouncer at a club.
The bouncer at a club.
Yeah. If the bouncer can't execute the mission and actually breach the uterine wall, it doesn't matter how great the VIP inside is, the party never happens.
I love that. Taking that bouncer analogy a step further, The mission that Bouncer has to execute is the single hardest physiological bottleneck in human reproduction, which is implantation.
Right.
The trophectoderm doesn't just sit there. It has to physically hack into the mother's system. It has to hatch out of that rigid zona pellucida shell. It has to secrete human chorionic gonadotropin, or HCG, so the mother's body realizes it is actually pregnant.
So it's very active.
Extremely active. And most importantly, it acts like an espionage agent, orchestrating a highly specific, localized immune suppression, so the mother's white blood cells don't attack the embryo as a foreign parasite.
Wow.
If that outer layer is deficient, the embryo cannot invade the endometrium, it won't implant. And at that point, the beautiful quality of the inner cell mass is entirely irrelevant.
That completely flips the script on what matters. And the 2023 guidelines take the subjectivity out of grading that bouncer, too. They don't want embryologists guessing if the outer layer looks cohesive anymore.
No more guessing.
Expansion is now a strict physical measurement. It has to be over 165 micrometers in diameter. And to get an A grade for the trophectoderm, the embryologist has to physically count exactly 14 or more cells visible on the equator of the embryo. Plus, the 2025 Istanbul update officially added a D grade to the scale.
Which is a massive relief for clinical labs, honestly.
Why is that?
Because previously, if an embryologist saw clearly necrotic, dead, or completely degenerating cells, they often had to lump them into the C category just because the grading matrix didn't have a basement.
Oh, that's frustrating.
Very. The D grade finally allows laboratories to decisively fail unviable embryos, rather than artificially inflating their quality just to fit a decades-old chart.
So, we have moved from treating embryo selection like an art form to using these incredibly detailed, mathematically strict systems, which naturally raises a huge question about the future. Can a machine do this better than a human?
Ah, AI.
Yes. The sources spent a lot of time on morphokinetics and artificial intelligence. We're talking about time-lapse technology, where instead of pulling the embryo out of the incubator once a day for a quick peek, cameras are recording continuous video.
And the shift to video is profound. We can now measure the exact velocity of development without disturbing the embryo's environment at all. We know the exact minute the maternal and paternal DNA merge. We know the exact hour it hits 4 cells.
And the computers can analyze that.
Yes. We are seeing the emergence of complex AI models that can analyze these kinetic variables and assign a continuous viability score, entirely removing the human eye from the equation.
But the sources also provide a really sobering reality check on AI. A major Cochrane review looked at all the data and concluded there is actually, quote, insufficient evidence that these multi-million-dollar time-lapse systems genuinely improve a patient's chances of taking home a baby compared to just using a standard incubator and a skilled human with a microscope.
Which is humbling, to say the least.
Yeah, the 2025 consensus explicitly treats AI as a helpful extra tool, not the ultimate decision maker.
The clinical restraint is necessary because AI models suffer from a foundational flaw. They are trained on human data. And as we discussed with the fragmentation issue, inter-observer variability remains a massive problem.
Right. Humans can't agree on the crumbs.
Exactly. Even highly experienced embryologists only agree on the grade of a fetal mass about a third of the time. If the humans feeding the grading data to the computer can't agree on what they're looking at, the AI's ability to predict a live birth hits a hard ceiling. It basically just inherits our subjectivity.
But beyond AI, there is an even larger caveat to this entire morphological system, isn't there?
Right, the ultimate trump card. Genetics.
Specifically, pre-implantation genetic testing, or PGTA. The data shows that while a beautifully graded, perfectly expanded blastocyst is statistically more likely to have the correct number of chromosomes, meaning it's haploid, it is absolutely not a guarantee.
Not at all.
The sources point out that in some patient groups, over half of the embryos with perfect visual morphology were actually genetically abnormal or aneuploid. They would never result in a healthy baby. So no matter how perfect the grading system is, whether you are using measuring cups or strict cell counts or a massive deep learning AI,
It doesn't matter.
grading an embryo visually without checking its DNA is basically judging a book by a mathematically perfect cover, right?
That is the defining limitation of modern embryology. Visual morphology shifts the probability. A good-looking embryo has a statistically better chance of being normal. But visual assessment has a very low diagnostic ceiling.
Right.
It cannot substitute for actual genetic testing, particularly for patients dealing with advanced maternal age or recurrent loss. We are rapidly approaching the biological limits of what merely looking at the outside of a cell can tell us about the genetic competence inside it.
So, to bring this all together, we have followed the embryo on a truly incredible 7-day journey today. We started with the frantic, dynamic dance of the day one zygotes, which proved too fast and chaotic to grade with a single photograph.
Yep, the blurry ballet.
Then we moved to the cleavage stage on days 2 and 3, where the field had to invent strict volumetric measuring cups just to stop humans from subjectively eyeballing cellular debris. We witnessed the genomic trust fall of the marula on day 4, where cells that couldn't turn on their own DNA were ruthlessly walled off and left to die.
Nature's quality control.
And finally, we arrived at the blastocyst stage, where we discovered the great plot twist. That the trophectoderm, the espionage agent that hacks the mother's immune system to form the placenta, is the true gatekeeper of life. It completely outranks the fetal cells themselves when it comes to predicting a live birth.
It is a staggering shift. We have moved from a subjective, observational art to a highly quantified predictive science, fundamentally changing how we understand the earliest moments of human development.
Which leaves you with one major thing to ponder as we wrap up. If non-invasive genetic testing, perhaps analyzing the DNA leaking into the fluid around the embryo, eventually reaches 100% accuracy.
Oh, wow.
Yeah, and if deep learning AI eventually perfects the visual grading, will the human art of embryology disappear entirely? Or will freeing up these highly trained scientists simply allow them to turn their microscopes toward the next great biological bottleneck in human reproduction? Thank you for joining us on this deep dive into the incredible microscopic world of the IVF lab. We'll catch you next time.
Leading innovation, fertility, excellence. Live by Dr. Pat. Leading innovation in fertility excellence.